So the plan for today, or the mission for today, is that I have half an hour to tell you what I did in the approximately last 5 years in terms of my work life. My work life mostly took place in the last 5 years here at the Zoological Institute, contained mostly within the building that you're also sitting in right now, while our wet lab working space is actually at the Institute of Biological and Chemical Systems at Campus North. To give you a bit of a frame of reference, where to place the things that I'm going to show you, I would mention at least my understanding of my strategic goals for the past 5 years. So the first one that probably sits on top of priority inside of my brain is the contribution to Großforschung, to large-scale research within Helmholtz, the POF strategy, and here the program Natural, Artificial, and Cognitive Information Processing. If that sounds a little bit difficult to understand, the theme here is to explore biological information processors with a view for next generation computer systems. So really build new types of computers for the future, which is also the main theme that you will find inside of my talk. Second, my mission was to bridge fundamental biological research that is running at a very good level at KIT with computer science and engineering that are also running at a world-leading level at KIT. And third is also to introduce computational approaches into research and also importantly the teaching portfolio at KIT in terms of its biology section. This as a frame of reference. Now I would, in a sense, start the talk by giving a motivation of what we do here. So oftentimes when I think of biology, I also think of codes and digital data. And you can see that code-based biotechnology is changing rapidly the world that we live in, making a huge impact. So you can, for example, see that current clinical practice for treatment of cancer has the option of having programmable immune cells that can target specific recognition molecular codes on the surface of cancer cells, allowing patient and tumor-specific treatment on the basis of recognition codes. You can also see that even clinical gene editing is making its first steps, so using options like CRISPR-Cas9 for correcting problems with specific genes, of course, leading also to huge ethical problems like germline editing that I think should not take place, but has taken place. I think the point where no one can argue that this has massively impacted our reality that we live in is the vaccine against the COVID-19 virus, which was digitally developed within 2 weeks and then sent to an RNA printer. And I think this is ample evidence that code-based biotechnology is becoming quite important for the world that we live in. You can go quite a step further, which some might consider still a fantasy world, and it's to talk about using DNA as a native biological hardware for actual computation. So computation as Nick is using his computer right now. I don't think it's a fairy tale anymore. This paper came out just before I started my professorship, and for me has been really impactful in how I think about the research program that we carry out. You can see here that actually still frames of a video have been encoded, written into the genome of living bacteria, and were subsequently retrieved by genome sequencing again. And you can actually find digital video frames out of living cells in this way. You might find this fancy. There have been further developments that I think become more specific. So for example, here, actual logical circuitry as needed for a digital computer has been implemented using DNA technology. And even further, there is now the possibility to detect different cancer markers from different cell types and logically integrate them into a DNA circuit that processes this information inside of living cells. Nevertheless, there are definitely fundamental challenges that you can understand if you think about the state that DNA has to be in when it is being accessed by other molecules, which is in aqueous solution. Aqueous solution means that information between molecules must be transferred by diffusion. Diffusion is very slow compared to the transfer of electronic charges inside of a real or conventional computer as we know it. So it is much slower. At the same time, the information transfer must happen by single molecule interactions, which are inherently noisy and stochastic. A conventional computer relies on the principle of being deterministic. So there is a very fundamental contradiction here. These contradictions to me are so fundamental that I think it would be assuming that I know the answer to these questions And I would rather go to a system that knows the answer and we can learn from that system, which takes me to the rationale of a research program, which is that we explore the architecture of DNA sequence readout, so the access to the information that is saved in the genome inside of the nuclei of biological cells. Because biological cells do use the genetic information quite reliably, they work, and this means that they must have in some way solved the problems that I just presented to you. So instead of trying to solve them just by ourselves, we go there to learn. And I think this would still be considered a normal classical biology program. And the crucial addition for what we do at KIT here is that we use this as an actual inspiration for architectures of future DNA hardware components, and not just as words that reference this, but we actually try to do this. To make this somewhat graspable, I divided the talk and also a research program in 3 main areas. They, of course, overlap, but what this mostly is divided into is that we have here the segment called observation. So there we use stem cells inside of zebrafish embryos and also cultured stem cells, and mostly methods of fluorescence microscopy to look at these architectures in very much detail and with a very high time resolution to try to understand what's going on there. Abstraction from these observations to more general principles is carried out using image bioinformatics, biophysics concepts and models, and of course, large-scale computer simulations. The third section would be reimplementation, to actually take what we have learned and build from it and upon it, where we use synthetic DNA nanomotifs to approach something I would maybe call lifelike architectures, and hopefully in the long run, this is why it's great. We can also use simulations to guide our design here. And this now actually allows us to understand the title of the talk, going from natural architectures to artificial architectures that carry out DNA-based information processing. The first section, observation. I would like to introduce 2 entities that are quite important in our biological research. The first is, of course, the zebrafish embryo, which is very easily accessible to microscopy because it's externally developing after the egg has been laid, and it's transparent and it develops very quickly. So within 4 hours, you have about 4,000 pluripotent stem cells inside of a living embryo. So they're probably quite functional and reliable. In here, we apply oftentimes fluorescence microscopy. So you can see, for example, on top, the DNA channel where you can locate one of the nuclei inside one of the cells inside of one of these embryos. And in the second color channel on the bottom, you can see RNA polymerase II. RNA polymerase II is the enzyme that reads the genetic code information and produces messenger RNA that the cell can work with to establish its function based on this genetic code. And this RNA polymerase II, or short POL2, is inside here, as you can appreciate, actually not homogeneously distributed, but organized within so-called transcription factories where things are centralized. You can see this in more detail if you apply super resolution microscopy like the STED approach here, which inside in the Nienhaus lab, who we collaborate with, has been improved by a double depletion approach. So you can see in even more detail how these factories here shown in this box are organized and really exhibit internal structure and quite complex shapes inside of them. So here, this is the second entity. Transcription factories will more or less carry us through the entire talk as a concept. I should mention here, there is a couple more related papers. If I would have had time, I could have added another one in minor revisions this morning. So I would like to give a nod here to the Nienhaus lab, reliable long-term collaborators, KCOP, Karlsruhe Center for Optics and Photonics, where we also use a STED microscope. And of course, also the zebrafish and Medaka facility, or animal house, which is central to our work. Transcription factories. To explain this a little bit more, they form at genomic regions called super enhancers, which are so-called control regions of the genome, here labeled in blue. And at these control regions, clusters with high concentrations of molecules that are part of the transcriptional machinery are formed. Into these clusters, you can potentially recruit genes that interact with these clusters and are regulated. Some genes go in, others don't, as you can see here. And genes that do not engage frequently have low or basically no levels of transcription, while genes that do engage can be turned on and become transcriptionally active, as indicated here. I said this is central to the talk. It's central to our work as well. So there is already some works that have been published by a group in the last years on this type of structure, largely enabled through our collaborators. So the Zabur Daev group, Mathematics and Life Sciences in Erlangen, Hiroshi Kimura's group at Tokyo Tech for labeling techniques, Silvia Erhardt here at the Zoological Institute, Camillo Ferrai from the Medical Center in Göttingen, just to name the people who make this work possible with us. And I would like to show just one highlight of one of the publications from this area, which actually shows you how we could reconstruct by a pseudo-time computational approach how a gene visits such a transcription factory. So you see in this one image, I will explain what you see in images that will follow. You see labeled in magenta, the recruited RNA polymerase II. This is the blob in the center of this image. And you can see labeled in green the embryonically expressed gene KLF2B inside of zebrafish embryos. And this is labeled by fluorescence in situ hybridization. And this is labeled in green. And you can see in both the algorithm detected the center of mass of both objects and draws a line where they are connected. And using this information, we can actually take still images, single images, and sort them in time. So you can see this coordinate S is continuously increasing. And you can also see how the gene is coming closer and closer to the factory that's placed in the middle. If we carry on with this for some time, you can see that the gene actually engages with the factory, kind of melts inside of it, becomes immersed. And as it is transcriptionally activated, you can see that the gene departs in the process, really heavily perturbing the structure or organization of this factory. And only as the gene has gone basically to the beginning of the process, the factory relaxes and becomes nicely roundly shaped again. These are example images. You can see that we can actually take hundreds of observations of one of these experiments, sort them, and get quantitative data out of there. So you can really see the approach of the gene in the first plot coming to very close distance to the factory. You can see in the second plot on the second color channel how the gene is first being induced and recruited. And in the third color channel, there's the actual transcription activity that a bit later on gets turned up as the gene becomes activated. You can read about this in more detail if you care about the more technical aspects, or you can look at these pictures where you can see the gene associating with the factory, being activated, and after the activation, leaving the factory, and then actually becoming transcribed and turning into mRNA. All right. Again, key collaborations here. Anne Koziolek from Computer Science is on the paper as a PI as well. Hamide Hajiabadi and Irina Mamontova-Nauwarta are also on here. LSDF to store all of the data that we produce here. It's quite massive. And of course, the InstantSim high-speed super resolution microscope depicted here, which was enabled by funding from the Hector Foundation and is really central as an instrument in our work. Okay, so to switch from understanding biology to thinking about computers, I want to introduce here something called the von Neumann CPU architecture. It consists of two main components inside of an electronic computer or digital computer. You have a central processing unit on the one hand side, and divided from this, you have memory or random access memory. The cool thing behind this is that inside of the central processing unit, you can put things that are working. So arithmetic, logic, really how you process the data. And you can divide this from where you store various pieces of data inside of your memory at different blocks. The trick now is that you can take your centralized processing unit and dynamically and selectively connect it to different blocks in the memory. In this way, actually allowing you to use one single processing unit to efficiently deal with a very vast array of memory, and also to give your computer a controlled state of operation. Control is the other important word. You need a control unit that drives the arithmetic logic unit and also tells it where to connect, carrying out this connection process. And now just by visual and structural similarity, you can already see how many of these aspects are actually mirrored in this transcription factory, where specific genes are pulled into one central factory that has the processing machinery to process the information that is contained inside of the genes and read it selectively. All right, so future research directions, we stay a little bit more in the area of biology for now. So we have here a project that is underway to see which RNA binding proteins are actually involved in the processes that we talk about. And here in mouse embryonic stem cells with Bao, who's sitting in front, funded by the Sino-German Projects Program of DFG. Another DFG project is to see how this type of spatial regulation contributes to embryonic mesendoderm specification. This is also underway. And last but not least, we found some really crazy data how nuclear actin networks might actually transport genes towards the factories at a higher speed than otherwise possible. Here, key collaborations, Xiaohua Shen in Beijing at Tsinghua, Martin Bastmeier for the stem cells, Uli Nienhaus for the imaging, and Robert Grosse in Freiburg for the actin detection. Also to be mentioned, research-based teaching. In this part, systems biology, you can see how we have fun doing the practical teaching. Here in the master's practicals, methods of developmental genetics, molecular biology of the cell. There is the bachelor's and master's combined literature seminar where I am one of the instructors. The other one is sitting there. And also international cell physics master in Strasbourg, where I am one of the international lecturers. All right, so this is observation, the first part. You can see here the number of papers. It's a talk where I should give review. So here's the list of papers. And we have in total 3 externally funded PhD positions from DFG in this section. This takes us to the next part, which is abstraction. Learn and generalize and build models from what we observe. And what I would do here is to go back to the same example that we had in the microscopy approaches before, which is the interaction of genes or their promoters with these transcription factories. So what we have here is polymer simulations of enhancer-promoter interactions in something that we call the shoebox model. The shoebox contains, tethered to the left side of the shoebox, a little piece of a chromosome that contains a super enhancer, as introduced in the beginning. And on the right-hand side, apologies to the online people, on the right-hand side contains a gene that is equally tethered with a small piece of chromosome. These are now put into not just a drawing and a sketch, but actually into code. So here we have the outside code that controls the overall simulation, keeps track of the chemical transitions that are needed to grasp this transcription control inside of a Python wrapper that whenever the chemical reaction network has been evaluated, passes this to a highly optimized LUMPS polymer dynamics library where we implement the structure of chromosomes with 10 kb resolution of the chromatin, which are connected into a chain using harmonic bonds. Here is an impression of what this looks like. Hilbert is also attached. You can see on the left-hand side the factory. On the right-hand side, you see the gene, its promoters, and you also see in red the small particles that are the transcriptional machinery. This work was carried out by Roshan Pritzak, formerly a postdoc with her group, now computational scientist at Solgate, using many of the skills that he developed here at KIT. In any case, so I showed the introduction of the simulation in an image. I think it really becomes alive and understandable when you see a video outtake of the simulation. So this runs at about 3 times real life speed. And let's let it run. You see on the left-hand side now the super enhancer, which has this cluster associated with it. In addition, you see on the right-hand side, thanks, Lennart. Yes, so you see on the right-hand side the gene with its gene promoter in blue. And in red, you see the inactive gene body, which right now is actually transcribed or active. It's going to turn red in just a moment. I hope. Yes, there it is red. And you can actually see how the gene repeatedly visits this transcription factory, turns from red to gray as it's active, and it's being expelled from the factory again after activation. So I think here you can see how these kinds of simulations really directly capture what we have seen in the experimental data, giving us an abstracted picture of these kinds of processes. These kinds of models also deliver concepts that we can hopefully exploit in the construction of these artificial DNA-based computing hardware units. And the concept that I want to introduce here is so-called surface condensation. Here is another type of simulation. It's called a lattice simulation. Again, you have red particles that correspond to the same red particles you have seen just before. And you see how going from the left to the right-hand side of these three simulations, the affinity between these particles is increased in the simulation. And only at the very highest affinity, you can see that the system undergoes something called liquid-liquid phase separation when you see a red droplet that is forming. This is without the presence of a polymer surface. With a polymer surface, you can see that even at lower affinities, you can have the formation of associations of these components or proteins or monomers. And the left-hand side, you basically have a bit of a coating on the polymer, so it doesn't give you all that much of a cluster. But in the middle, you have surface condensation here. And you can see that we have supported by the polymer forming specifically at the polymer the growth and formation of size-limited condensates. Yeah, you can read about this here. So what this gives us is, in addition to what we have had in the picture of the transcription factories before, where we merely see that genes do associate or do not associate, we can now understand how liquid phases that code genes and enhancers make this possible. So, for example, the liquid phase 1, shown in green here, is supposed to be not compatible and not miscible with the red phase. Same for the liquid phase 3. So these genes will tend not to mix or not associate into the transcription factory differently. The liquid phase 2 at the super enhancer at high concentration is the same phase as associated with this gene number 2. And this leads to the ability to associate with this cluster in a selective fashion. So we now have a mechanism that can drive this. Also that we can construct because we have a model. All right, so in this section, future research directions include here that we take this idea of taking still images and combining them to discover, with the help of AI or deep learning, discover dynamic processes and reconstruct them from these still images. Niels is also sitting here in the front, student in the HITS for Health graduate school. And we also want to take simulations like here, not just shoeboxes, but whole genomes that we simulate into inside entire nuclei and lift them to the to-be-formed DNA unit of the virtual materials infrastructure within the new Carl Zeiss Center Synthetic Genomics that's coming up next year. Collaborations here would be Wolfgang Wenzel and importantly Mariana Kozowska at INT, and this to-be-formed DNA unit that I would like to mention here. Modeling and simulations, there are a number of papers that are really exciting that we worked on, but they don't fit the main narrative. So here are in total 4 of them that model things like myosins working on actin and oscillatory systems, hyphae of fungi finding each other because they oscillate in an anti-synchronous fashion. So really exciting work, but no time. Most of it done actually together with Ralf Mikut at EIE and with their biological collaborators, both in the room, Reinhard Fischer, Nick Folks here in front. And we use BW Unicluster for the heavy lifting of the computation that is needed. Research-based teaching. Here in terms of computational skills, you can see I'm having fun. Whether the students have fun or not, you decide or you ask them. There's the image processing bioinformatics section of Methodenpraktikum at the bachelor's level. At the master's level, we have Methodenpraktikum Mikroskopie, where I contribute image processing. And we have the newly generated 4-week course Bioinformatic, where I do 1 full week of image analysis with MATLAB, as you see here. And there's the BIF IGS graduate school, where I lead the 1-week course Python application and contribute 1 of the days of that course. Again, to take stock here, that's the publications and preprints from that time. And there were in total 3 co-supervised PhD positions within the HITS for Health graduate school. Okay, so we have seen, we have understood, can we build? Here, our method of choice for building some of the things that we think we have understood is to use so-called synthetic DNA nanomotifs. They consist of something that most of you know as primers for your PCR. So it's short DNA oligomers, as you see on the left, below 100 nucleotides, super cheap, delivered usually within 3 days, then maybe 1 week within goods in. Anyway, these can be annealed if they were designed in the right way previously into these sort of metastructures or nanomotifs, as you see on the right-hand side. And importantly, they have sticky ends that are short and single-stranded. They can have sequence-specific interactions by homology and complementarity, but they only have short-lived interactions. So they can be very transient and give you things like liquid phases or association with targets, but transient associations. So what becomes possible is to rebuild some of the structures that we have seen before. So you see, for example, that nanomotifs transiently interact with other nanomotifs via the palindromic sticky ends, forming little condensates. And if you add a fourth non-palindromic sticky end that targets specific parts of a previously synthesized strand labeled in green here, you can even localize and assist the formation of these condensates to specific parts of a DNA strand that you can sequence program. And this is not just fantasy. We have data. So you can see here in purple or magenta in these pictures, the nanomotifs. On the left hand, actually no nanomotifs. In the middle, you have surface condensation. The picture shows white, which means purple and green is together. We have condensation on the surface. And on the right-hand side, you see normal phase separation. This doesn't happen when you don't have the surface present. So you can see that if you don't have the surface present, there is actually no condensation. You need the surface to assist the condensation, except you go to higher concentrations where you have rather non-target specific phase separation that proceeds surface present or not present. So again, we have established this regime of surface condensation that targets your condensates to specific parts of DNA strands, actually mimicking some of the crucial aspects of transcription factory organization. All right, we're actually nearing the 10:30 mark. We started a few minutes after, so I also only have a few minutes left. I should mention on this slide, these are experiments done in a bachelor thesis and a master's project internship. So this is, sure, research-based teaching, you can say this, but I think here the teaching is the research, is the teaching. The same applies for simulations that, because of the limit of time, I apologize, Aaron, I cannot show, but these were also carried out in another F3 practical together with supervision by our PhD student, Aaron, who's sitting here in front. So again, research-based teaching, I think it goes a bit further. It really drives our research. Key collaboration to make this possible is Kerstin Göpfrich in Heidelberg. Some of you might know her. And while this is teaching in the context of theses and internships, it turns into solid papers, as you can see here. All right, to close off here, future steps towards DNA hardware. So this is how sometimes I picture how we will get to something like this von Neumann architecture. We need a CPU droplet that is one droplet species. And then we want to connect this to various droplet species here, 1 to 3 or maybe n species. And how we can do this is to actually use bivalent connector DNA nanomotifs that have a valency for the CPU droplet and for one of these target droplets, leading then to the targeted fusion of the red and the blue droplet, giving the machinery fused to the target surface that contains your genetic information or DNA sequence information. And in this way, we can reconstruct the same structure as we know it from electronic hardware. Future steps, we need more memory addresses than just 3 or 2 as we have currently. And there we need something called simulation-based digital twin design and also more complex DNA strands to make this possible. Maybe we don't actually need these connector motifs, but we can induce a fusion of specific CPU droplets with specific memory strands using light or flux microscopy with Moritz Kreising, who can actually move particles and droplets and other objects inside of a liquid just by the use of light. And in this way, maybe we can do something like quantized chemistry using droplets. Of course, in the long run, we actually want these things to compute. So we need to read and write and edit. And for this, we should add components such as base editing enzymes, replication transcription enzymes. And one of the major approaches in the field for logical operations is actually toehold-based strand displacement, or some people might know this as unzipping and zipping DNA reactions. The crucial planned collaborations and planned infrastructures for this, again, the DNA unit of the virtual materials infrastructure here at KIT, and the planned extension of the DNA synthesis facility at Uni Heidelberg in the framework of the SAIS Center Synthetic Genomics. Of course, Moritz Kreising, who recently came to KIT. And we're also talking in terms of these components that should be added here to some of the people who know most about this at KIT, which is Christoph Niemeyer's group, and specifically his colleague Kersten Rabe in his unit. All right, with this, we complete the picture. Here's a few papers, more papers in this section than I ever expected during my tenure-track time. It went faster than I thought. We established a new focus field within the Martellis Center, which is self-assembled, adaptive, and biomimetic material systems, where I am co-PI. And I'm also co-PI of the application for this Carl Zeiss Center. I'm not going to say the name again, but yeah, the funding has been awarded and the work should start the next year. One of the main responsible surveyors is sitting in the back of the room. Yes, and of course, especially this last area, topically, largely contributes to this theme of life science engineering. If that wasn't clear, I just want to mention it. This closes off. So thank you everyone who dares to work with me and doesn't immediately run away. So current group members, I give a nod. Past group members have done a lot of work and have gone to bigger and probably better things. And of course, the collaborators. This is super interdisciplinary. For most of the things, I have ideas and I need help and I get it and then it works. And of course, the hidden hands on the ground, fish facility, technical staff, administrative staff, and of course also section 1, KIT service units. We curse a lot, but in the end things get done. So it wouldn't be possible without it. 1034, that's on